Hi, today I'm going to summarize the last class, which was on systems biology, which was a little bit difficult, but today I'm going to summarize in the easy way. So here, system biology uses the programming mathematical equations to create computational models of the complex biological systems, moving beyond a reductionist view to understand how components like genes and proteins interact to produce emergent properties and functions. These models integrate high throughput experimental data, genomics, proteomics, and metabolomics to analyze the system dynamics, enabling insights into disease, drug discovery, and synthetic biology. Key tools which were previously used, for example, SPML, simulation languages, supported by programming libraries and platforms such as MATLAB, Python, and some more specialized packages. The right side figure is representing in the picture, that is basic knowledge of biology, computational data science, and mathematics, biophysics, bioengineering are required. And then you will be much more comfortable in systems biology. And then if you are going towards the better understanding, then you need basic biological knowledge or understanding, data and hypothesis, wet lab experiment, then dry lab experiment, system analysis and theory formation, predictions, experimental design, and experimental data analysis, and finally, the final conclusion in with respect to the realistic biological systems. Then, what are the mathematical equations which in general you are required? For example, this is a small module or a small component of the signaling pathway. Then there are three reactions which you can see here. And then if you are going to make on the paper or computer, such equations will appear for each component of this small signaling pathway or pathway component. In the similar way, you can divide it into models, modules, motifs, macros, rules, equations. So you need to also set the rules based on some biological systems. And this is the same thing which basically is displaying that what are the information which you need to have before you proceed to enter into systems biology realistic approach. Then here I have given my own data. This is one of the network which is basically merged for four path, pathways: AKT, JAK-STAT signaling, TGF beta, and Hippo signaling pathways. And these genes were differentially expressed in realistic data in case of a specific cancer. And what we have done it, we took all the genes or proteins which were observed in our data as remarkably changed and then merged them. And then we observed that the different proteins have different kind of combinations or interactions with each other. And in the same way, they will also integrate with biological functions. So those genes which are having more connectivities, they will be basically more significant connectivities and followed by the connection with the biological pathways. They will have more importance. And that's why, in general, when the people are trying to understand what drug target purpose in human disease, they target or try to find the drugs for targeting that specific protein. Then, how it works? Basically, you need to proceed with several steps, and then you go for experiment, modeling, and analysis, and finally enter into realistic application. In the same way, you can think about a system or more system in a human. Then you can make a model. So basically, create a small module and then expand the modules by integrating the small modules into one model as a bigger model. Then it will mimic the system where you can integrate more than one organ or more than one problem. Now, what are the comprehensive measurement? For example, here I have discussed earlier in comprehensive module, a technology or understanding. For example, this is FGFR4 pathway. Then here I have shown what are the downstream signaling pathways which could be regulated by the change in FGFR4. So one pathway Jack, then MAP kinase pathway, and then AKT pathways. So this is AKT pathway, this is MAP kinase pathway, and they are connected with each other. That is called pathway crosstalks. So here you need to create such kind of map first and then prepare the model or coding and set up the final model for simulating the entire system. And then you move to one step and then from one step to another step. Next, it comes to impact of biology in drug design. So this is what it will show you if you will model them in addition to NGS or gene expression or mutation or some epigenomic profiling or proteomics profiling. Then you need to also understand the dynamic behavior of the protein when you are going to add some new drugs, then how they will behave before and after the drug involvement. Then there are some other technologies which are involved. This I have discussed a lot. So here there is another way of master regulator proteins. For example, these two networks are the same network for prostate cancer. So these are the master regulator. For example, TRA2A, TRA2A, one protein, then NOL4, then CLN5, then this is STK38, then another RNF5. Then another one is basically there are less connectivities, but their positioning is very important. They are connecting two different clusters of the proteins. So here, for example, RANV2 and then SERA, then NRF1. So these are also crucial in terms of if there is any kind of disruption in these proteins also, then there will be huge change in terms of functions also. So these are the master regulators, key regulator, junction proteins. Oh, junction, I'm not talking about in biological terms, but in terms of regulatory functions. So they are connected or connector protein. So if there is some differences, they could lead to huge alteration or impact in terms of vital function. Now it comes to structural ability, stability. So whenever you are designing the model, you need to see, check the robustness. When you are using the hypothetical parameters and when you are using the realistic parameter, then how much these systems are robust. So then it will also help you to understand the impact of changes as a result of treating the patient with drugs. Now, this is one of the examples to show the real application and how the system behaves. For example, here you could see this is one case for type 2 diabetes. So, Rho A GTP, that is activated Rho A, how they could behave. This is one of the model to check computationally how the Rho A activation works in case of diabetes and normal cells. And this was the example for other things, like, for example, bacterial chemotaxis, but this is the example for human or mice model or animal model. So this is the experimental data, and this is the mathematical model. So your model should behave in the similar way as the experiment or realistic signaling behave. And for this purpose, you need to design your computational model in a proper way, and the modular design must be really compact and without redundancies. So here you need to focus on the components and devices, modules, and finally the system, how the system could behave. And this is the example. So earlier it was a model which we prepared. Now it is the example. So before and after the treatment with activated protein A, activated protein PC, APC. So this is when the mice model has diabetic condition, and this is normal or healthy case. So in experiment, we could see the activation of row A is continuous or persistent. That is also called sustained response. So signaling is on and it remains on. While in case of normal, there is signaling at a specific time point, that is 10 minutes. And in the same way, we could see in our computational model, the signaling is continuous, and here the signaling is completely transient. That is in the form of a peak. And this the difference in the model is this part is not working properly in case of type 2 diabetes. So here we could say alpha 5 beta 3 protein is not working. That means it is absent in case of diabetic condition. And here you can see the amount of dose or dose amount for the drug. When you are increasing the dose amount, still the output response remains the same. And this comes under robustness. So how robust the result is. Now you can understand the Systeome project. So this is from one of the review paper, Nature Review, and you can see also here you have genomic level information from high throughput data, SNP, CNV, LOH, loss of heterozygosity, copy number variation, genomic rearrangement, rare variants. Then epigenomic data at second level, that is epigenomic data, DNA methylation, histone modification, chromatin, accessibility, transcription factor binding, and miRNAs. Then it comes to transcriptome level, gene expression, alternative splicing, a long LNC RNA or long non-coding RNA, a small RNA. Then at you come to the proteomics level, then you need to have the proteomics expression, PTM, post-translational modification, cytokine array, and then metabolome. Here, metabolite profiling in serum, plasma, urine, CSF. And you, as a system biologist, you are not just uh, analyzing the individual level information only, but also you need to integrate them in a model in order to mimic the entire system. So you need to integrate all the different level of information into one model to show the final phenotype of this organism or the organ or the system. Now, for example, if you have nutritional processes, and you have the data for cancer initiation, progression, metastasis, then you can just go for analysis as a bioinformatician for at each level. But if you are a good system biologist, you will be able to integrate them from cancer initiation to progression and metastasis. Then you could be able to find the realistic reason behind the cancer initiation, progression, or metastasis. In the same way, if you have gene expression data, then how can you proceed in addition to gene expression, enrichment analysis, pathway enrichment analysis, and network level understanding? You need to integrate all these information, and then you work as a, or you are considered as a system biologist. Now it comes to mutational data or CGH data or whole genome sequencing data. In the same way it works. Now here, this is another example. If your mutation is a specific kind of mutation, for example, dominated by C2T. So C is mutation, and C mutation, C is mutated with T, or C is mutated with A, or some other combinations. So there are 12 possible combinations. Then how could you integrate them? So one way is to analyze all the information, what kind of mutation is more dominant, and then how to integrate them into a system, the impact of each type of dominated mutation. So here, this is summarized way for a complex system biologist, how you can work. Data collection, collect all the data in a purified way, and then go for, if you want to integrate some kind of a structural biology things, then apply computational chemistry, then apply system biology approach. For this, you need to have a design, synthesis of a small molecules also, immunohistochemistry, and final conclusion. Then it comes another way. Final model is represented here in the form of a complex system, how can you correlate it with all the hallmarks of cancer? This is what you need to think in system biology or find the solution. How can you find the exact inference of all these multi-level information into the prediction of hallmarks of cancer? Finally, thank you very much. Please don't forget to subscribe our channel. In the next class, I will be discussing very interesting topic, that is basic concepts in medical systems biology. Thank you very much.